Hi, my topic today is non-disjunction. So non-disjunction is what is it's called when the chromosomes during meiosis don't separate evenly. So they that can happen, it says, during either one division of meiosis. So this can happen in meiosis 1 or it can happen in meiosis 2. And I want to go through the difference in the results of what happens if it happens in meiosis 1 or 2. So the result could be a sperm or an egg, so a gamete that either has an extra chromosome or one that lacks a chromosome. So when that sex cell that has gone through meiosis improperly combines with one from the opposite sex, the resulting fetus will have cells with an extra chromosome or will be missing a chromosome. So generally, such fetuses will not develop properly, and we would say that the baby would have a birth defect. Okay, so what you're looking at here is a karyotype. So a karyotype is just a way of studying the chromosomes in a cell, because obviously they don't line up in a cell in order from largest to smallest. But during metaphase 1, scientists could take a picture and spread them out and have a look at them in order like this. So we know that these are like an address now, and they're lined up from largest to smallest. And so we know what information is found, say, on this set of chromosomes or this set of chromosomes. So we've got that mapped out. And so scientists know what's going on each one of these chromosomes. So they're like it's just like it's like a little address. So we could talk about the 8th set or the 4th set or the 21st set. And then this last set here, these are going to be the sex chromosomes. All the rest of them are autosomes, and then this is the sex chromosomes here. And we can see that this is a karyotype for a male person because they have an XY chromosome. Okay, so non-disjunction could technically happen on any of these sets of chromosomes. It could happen, but probably the probability is that there would be no live birth resulting from that, because these are not compatible with life if it happens on one of these big important chromosomes. If it happens on a smaller chromosome, then there are certain conditions that are associated with having an extra chromosome or a chromosome missing, and conditions and syndromes and things like that. And we'll go through a couple of those. A lot of, well, I guess one of the conditions or the syndromes that we know about, most people know about, is Down syndrome. And that's a result of having an extra chromosome on the 21st set. And so another name for Down syndrome is trisomy, which just means 3 chromosomes on the 21st set. Okay. And then there's quite a few conditions that happen on the sex chromosomes as well. And so if for example, a girl was only born with one X chromosome and not the other one, she would have Turner syndrome. If a girl had an extra X chromosome, she would just have triple X, and she probably wouldn't even know she had it. It's not really associated with any symptoms. And then there's XXY male who would have Klinefelter syndrome. But anyways, we'll look at a few of those syndromes afterwards. So just know that it can happen, and if it's going to happen on one of these smaller chromosome sets, then that's compatible with life, particularly on the sex chromosomes. Right in the way. Okay, so I wanted to just review before we did that. Normal meiosis. So what's happening here, let's start up here. And as you know, this cell has gone through interphase and has doubled up all of its chromosomes, and you can see that that has happened to there. And now it's going to go through its first meiotic division, and we're splitting those chromosomes. They're still attached to their partners, that they're still in partners, I guess, there. Then meiosis 2 is going to happen, and we're going to split those up. And what we're going to end up with here is a cell with half of the genetic material of the original cell. Okay, so these in humans, N equals 23. So these gametes, whether they're eggs or sperm, all have 23 chromosomes, and that's normal. So if meiosis happens in meiosis 1, in the first meiotic division, here's what this might look like. So I tried to draw in the spindle here. So this is metaphase 1 here going to happen right away. So when that happened, you can see the spindle reached out and it grabbed an extra chromosome here, which it shouldn't have. The spindle from this side should have grabbed that guy, but he didn't. So that's the oops. And now when they divide, you see what happens. We've got this chromosome on, we've got too many chromosomes on this side, and we should have had that green one up here too, but we don't. So now when it goes through meiosis 2, look what happens. We've got an extra chromosome here, an extra chromosome here, and we're missing one here, and we're missing one here. So we've got n plus 1, n plus 1, n minus 1, and n minus 1. So when non-disjunction happens in meiosis 1, you do not get any normal gametes whatsoever. So none of these gametes are normal. They all have an abnormal number of chromosomes, whether they're egg or sperm, it doesn't matter. Okay, so now let's just contrast that with my non-disjunction in meiosis 2. So we start again. We've already got our doubling has already happened here. And so he's going to divide the first meiotic division, and all good so far. This happened normally now. Now, when we get to meiosis 2, now here, this side has done what it's supposed to do, and we've got half of the genetic material here, which is good. But here, we've got a oops, and that happened in metaphase 2 this time. So on this side, we've got this one, and I'm going to zoom in. I've got this on another slide. But the spindle reached out and it grabbed this whole thing, and it was supposed to pull them apart, and it didn't. But the spindle pulled the green one apart, which was good, but both of these purple guys ended up in this gamete. So now we have n plus 1. So in a human cell, that would be 23 plus 1 extra. And in this side, we've got n minus 1. But look on this side of the equation. These ones are good. They're n and n. So if non-disjunction happens in meiosis 2, then we're fine. We're 50% fine anyways. We can have 2 normal gametes and 2 abnormal gametes. So just to zoom in a little bit here. Here's what happened in metaphase 2, and then anaphase pulled them apart, 2 opposite poles. So on this side, we've just got the 2 purples and the green, and on this side, the green. So that didn't work out very good. So now we can kind of look at what happens during fertilization here. So And this would be two normal gametes here, egg and sperm, and they're both got 23 chromosomes, and that goes back into 46 chromosomes, which is the normal number in human cells. Now on this side, we've got one gamete that has an extra chromosome because of non-disjunction. And this gamete has a normal number of chromosomes. So what we're going to end up with is an extra chromosome, and that resulting fertilized egg or zygote will have 47 chromosomes. And the same here, I just switched it around so that you don't think that it's always the egg. It could be just as easily be the sperm. Okay, and that's going to have 47 chromosomes here. But on this side, we've got N minus 1. So this represents a gamete. And again, it can be egg or sperm. It doesn't really matter. That is missing a chromosome. And this would be a normal gamete. You can't really see it, but it says 45 chromosomes. So this one's going to have 45 chromosomes, and when it should have 46. So that's non-disjunction. I hope that makes sense. We'll go through in your textbook a little bit more about some of the different conditions that are associated with non-disjunction, particularly in the sex chromosomes. And that's all I have for you right now.